When you see a perfect apple in the store or hear about corn that can survive a brutal drought, that's not an accident. It's a story of incredible patience, of science, and of time. Today, we're going to dive into one of those stories, a 15-year mission to build a better, stronger wheat, one single gene at a time.
So our story really begins with two main characters, two very, very different kinds of wheat. One is, well, it's our champion, the one everyone wants to grow. But this champion has a secret, a critical weakness that puts everything at risk.
Okay, so meet variety A. Think of it as the star quarterback. Everybody loves it because it gives you a massive harvest, a fantastic yield. But it has one huge problem. It gets completely wiped out by a common disease called rust. Then you've got variety B. This one's kind of the wild cousin. It doesn't produce nearly as much grain, but it has a superpower. It's naturally tolerant to that same exact rust. So the mission, if we choose to accept it, is to get the best of both worlds into one super plant. To pull this off, we have to talk genetics for a second. Every plant has its own unique genetic code, its genotype. Now, the gene that makes wheat susceptible to rust is dominant, so we'll call that a big R. The gene for tolerance, the one we want, is recessive, so we'll call that a little r. Our popular variety A is RR, and our tough-as-nails variety B is RR. So the million-dollar question is, how on earth do we get that little RR into our superstar RR plant? And this isn't just some casual experiment in a lab, This is more like a high-stakes genetic rescue mission. The goal is incredibly precise, the process is painstakingly long, but the payoff, well, the payoff could help secure food for millions of people.
So here it is, the game plan plain and simple. We need to transfer that one little gene for rust tolerance, the RR, from our wild plant into our big-shot, high-yield plant. And here's the real kicker. We have to do it without dragging along any of variety B's other traits, like its low yield. We only want the good stuff.
All right, year one. The journey kicks off with a simple cross-pollination. We take pollen from our popular parent, variety A, that's our RR, and we use it to fertilize our donor parent, variety B, our RR. This creates the very first hybrid, what scientists call the F1 generation. And the result of that first cross? Every single one of the new plants has the genotype RR. Now, because that big R for susceptibility is dominant, they're all still vulnerable to rust. So you might be thinking, wait, that seems like a step backward, but trust me, it's not. We've successfully snuck that tolerance gene into the DNA. Now the real work can begin.
Welcome to the heart of the operation, folks. This is the backcrossing cycle. It's basically a fancy way of saying rinse and repeat. This is where the true test of patience comes in. It's a slow, meticulous process of crossing our new hybrid back to that original popular parent over and over and over again.
So in year two, we take our hybrid plant, the RR, and we cross it back with our original popular parent, variety A, which is RR. This is the first backcross. What we're really doing here is trying to dilute all that genetic information from the wild parent, basically washing it out while holding on tight to that one gene we desperately want.
And when we do that, what do we get? Well, it's basically a 50-50 split. Half of the new plants are RR, just like the popular parent, and the other half are RR, still carrying our hidden tolerance gene. The problem? They all look exactly the same. You can't just look at them and see which ones are carrying our precious cargo.
Which brings us to year 3, where we do something really clever. We take all those plants from the last step and we let them pollinate themselves. See, by doing that, that recessive little RR gene finally gets a chance to show itself. We then expose all the plants to rust, and what happens? Only the ones that are tolerant, the ones with the RR genotype, survive. We found them. We found our gene.
Okay, that's a huge win. We've managed to isolate the plants that have the rust tolerance we're after. But, and this is a big but, genetically this plant is still about half that wild variety B. It's got the rust tolerance, yeah, but it probably also has the lower yield and other wild traits we don't want. So what do we do now?
You guessed it. The answer is simple, but it takes forever. We do it all over again. We take our new rust-tolerant R plant and cross it back to the popular R parent. Then we self-pollinate, we select the survivors, and we repeat the whole cycle again and again. Each time we do this, the new generation gets genetically closer and closer to being just like our popular parent, but it always keeps that one critical gene for rust tolerance.
So this whole cycle of backcrossing and selecting, this isn't something you do over a weekend.
Let's zoom out for a second and look at the true scale of this thing. This whole process is a masterclass in the long-term vision you need in agriculture.
Just look at this timeline. It shows the entire journey. That backcrossing and self-end cycle we just talked about, that gets repeated for 5 full generations. That alone takes us all the way to year 9. Each cycle is making the plant better and better, closer to our ideal. But even after almost a decade of work, we're still not done.
Right. So from year 10 all the way to year 14, our new and improved plant has to prove itself in the real world. I mean, all that work in the lab is great, but it means nothing if it can't perform out in a farmer's field. This means years of trials, first small ones, then massive, multi-location trials all over the country to make sure it's not just good, it's consistently great everywhere.
And then, finally, after a decade and a half of meticulous, patient work, we get to the payoff. The resilient harvest, the moment of truth. Was all of that effort, all of that time, worth it?
Oh, you bet it was. Look at this number. The final product, our brand new weed variety, is 99.9% genetically identical to that original, high-yield, super popular variety A. We've managed to keep everything that farmers and bakers loved about it.
But tucked away inside that 99.9%, we've installed that one tiny, little, unbelievably powerful trait. The result is exactly what we dreamed of all those years ago, a popular, high-yield wheat that is now completely immune to rust. It is the perfect blend of our 2 original wheats.
And this right here, this is why it all matters so much. This isn't just a cool science experiment. This kind of work protects our crops from being wiped out by disease. That means more stable food supplies for all of us and more security for farmers. Ultimately, it's one of the most important tools we have for feeding a growing world population. So this 15-year marathon, that's the classic way plant breeders have been doing this for decades. But it leaves us with a really fascinating question to chew on, right? Today we have these incredible new tools like CRISPR gene editing that can go into a plant's DNA and make a single precise change. So how might a tool like that change this incredible 15-year timeline? Could we one day build a better plant in a fraction of the time? That's something to think about.